In regular cell contraction, myosin cross-bridges bind to actin. Power stroke cycling occurs. The overall actin cytoskeleton contracts as a result of the synchronized power strokes. The sarcolemmas, that is, muscle cell membranes, also contract to maintain the integrity of the cell. Notice that the actin cytoskeleton is attached to the sarcolemma via integral membrane proteins such as dystrogylcan and, as a result, dystrophin. So then what happens if dystrogylcan is removed or doesn't function correctly? Well, in a normal cell, a cell growth signaling cascade is stopped by dystrogylcan. Although research is still ongoing, dystrogylcan seems to act much like a MAP kinase scaffold, but inhibits the cascade through incorrect lining up of proteins, or physical or allosteric blockage of the substrates or active sites. With dysfunctional or absent dystrogylcan, the signal cascade simply proceeds. The extracellular growth factor complex is activated by an outside signal. The guanine exchange factor part of the complex, son of sevenness, switches the GDP and RAS, shown here as an indented yellow protein, with a GTP, now shown here as a functional protein in a yellow sphere. RAS GTP attaches to RAF, a MAP kinase, activating it. RAF phosphorylates MEK, a MAP kinase, activating it. MEK phosphorylates MAP kinase, activating it, which then phosphorylates transcription factors, which affect the genetic material in the nucleus. As a result, the cell divides. So then what happens if you remove dystrophin instead of dystrogylcan? If dystrophin is completely removed, actin and myosin still contract, but the sarcolemma is no longer attached to the cytoskeleton. The sarcolemma therefore doesn't contract in sync with the actin and the myosin. As a result, the calcium channels embedded in the cell membrane open, causing stress and activation of calpain, shown here as an orange blob, a proteolytic enzyme that digests proteins needed for contractions, shown here by the removal of proteins. Eventually, the muscle becomes weak and eventually dies. Phosphokinase is an enzyme that converts creatine to photocreatine, dehydrolyzes ATP to form ADP, and is found mostly in muscle cells. When cells degenerate and break open, as shown in this video, creatine kinase spills out into the bloodstream, causing elevated levels. This is one major indicator of muscular dystrophy, a creatine kinase blood test that comes back positive for elevated levels.